Hi, I'm Alina Avanesyan. I'm a postdoctoral researcher at the Department of Entomology, University of Maryland. I work on ecology and evolution of novel plant-insect interactions. And today I'm presenting one of my projects. In this project, we looked at the plant responses to grasshopper herbivory and how they differ among different cultivars of the introduced Miscanthus sinensis. Miscanthus sinensis is a beautiful ornamental grass. It was first introduced in the US in 1893, and since then it's quickly established and spread across the state, having a very high invasive potential. Miscanthus sinensis has more than 100 cultivated varieties, and it's been shown that varieties with striped pattern are less vigorous and less invasive, while all green plants are more aggressive. And in our research, we are curious why is that and what mechanisms underlie such difference in invasive potential of this grass. Specifically, we are interested in one of such mechanisms, the interaction between introduced Miscanthus sinensis and its native insect herbivores, such as grasshoppers. Previously in our research, we showed that introduced grasses demonstrate low resistance to grasshopper herbivory than native grasses, while they tolerate the herbivory similar to native grasses. We also show that grasshoppers prefer to feed on introduced plants rather than on native plants. So for this project, we were interested whether different cultivars of Miscanthus sinensis differ in their resistance and tolerance to grasshopper herbivory. To explore this, we used Melanopolis grasshoppers, which are very common in our field area, and we conducted field experiments and greenhouse experiments. In our field experiments, we randomly planted 150 plants, 30 plants per cultivar, and we measured plant growth and leaf damage from grasshopper herbivory at 4 time points during the growth season. Here are our results. We found that plant resistance differ among cultivars, and one cultivar in particular, Morning Light, showed the lowest resistance in the beginning of the season, but the highest resistance at the end of the season. When we compared the tolerance between cultivars, we observed the same pattern. Plant tolerance differ among cultivars, but 2 cultivars, such as Grasshopper and Autumn Anthem, shows the highest tolerance among all the cultivars. Then we conducted the same experiment in the greenhouse. We used the same 5 cultivars, and we used 150 plants, 30 potted plants per plant. We randomly placed potted plants inside of the large enclosure, and we introduced 45 Melanopolis grasshoppers inside of this cage. We let grasshoppers feed on plants for 3 days, and then we took the plants out and we conducted the same measurements. We measured plant growth and leaf damage in the beginning of the experiments, at the end, and 2 weeks after the conclusion of the experiments to see how plants recover after leaf damage. And here are the results from our greenhouse experiment. We observed variation in plant responses to grasshopper herbivory. One cultivar, which is Autumn Anthem, showed particular lowest resistance to grasshopper herbivory, and one cultivar, which is Grasshopper, shows the highest tolerance to grasshopper herbivory. In conclusions, we observed that grasshoppers feed on all the cultivars in both field and greenhouse experiments. Also, plant responses differ among the cultivars. However, 3 cultivars in particular, Grasshopper, Morning Light, and Autumn Anthem, showed low plant resistance compared to other cultivars. It's also interesting in the field experiment, we observed that the lowest resistance of these cultivars in the beginning of the season changed to the highest resistance in the end of the season. Also, two cultivars, Grassolimus and Autumn Anthem, show significantly higher tolerance than other cultivars. Considering that all these cultivars are all green cultivars, our results suggest that these particular cultivars might have high potential for invasiveness. For our future research, we would like to take a look at the evolutionary changes of plant responses. And we want to compare the plant responses, which we observed for the cultivars, with the responses of Miscanthus sinensis wild type. With that, I would like to thank all my lab mates and all the people who helped us with planting and establishing the plots. Thank you very much. I'll be happy to take any questions.